Hello and welcome to this Pharmaron podcast, part of our DMPK Insights podcast series. My name is Simon Taylor. I lead the DMPK function in our UK-based Integrated Drug Discovery Department. Today, I'm delighted to be joined by Dr. Scott Summerfield.
Oh, hi Simon.
Before we get into the detail today, I'd like to provide an introduction to Scott. Scott is currently Senior Director of Pharmaceutical Metabolism at our Rushden site. He has a Ph.D. in protein mass spectrometry from the University of Warwick and has a 25-year career in pharma across SmithKline Beecham and then GSK across bioanalysis, biomarkers, DMPK project roles. And he has a specific interest in neuroscience DMPK. So Scott's with me today to discuss his recent publication in Pharmaceutical Research entitled Free Drug Theory, No Longer Just a Hypothesis. This was published in 2022 with GSK colleagues Dave Fairman and James Yates. And this subject matter continues to be an incredibly important topic in the DMPK community, with the topics fundamental to drug development and optimization. So I'm really looking forward to discussing the concepts today with you, Scott. So maybe I can start by asking you, I guess, what was the inspiration for this article? Why write it?
Well, it was an invited contribution to a Pharmaceutical Research Journal special edition, recognising the work of Margareta Hammarlund-Udenaes. Margareta, I really apologise for the pronunciation of your name, but Margareta solidified the idea of KPUU, distributional, constant and bound across the blood-brain barrier. I did a lot of work in neuroscience in the past, and so it was great to be asked to do that. So I wanted to write an article that was relevant, and one that was burning into my brain like a worm gnawing at it was one on the free drug hypothesis. So really the motivation was to kind of get that out of my mind. But I really struggled. 10 years that was sitting in my mind, and I just couldn't find a way to wiggle it out.
That's great. That's been done now. So I guess we should probably start with one of the fundamental questions, I guess, for people listening to this is, what is the free drug theory?
So we defined it as the unbound concentration of drug at the receptor biophase that's responsible for eliciting the pharmacological effect. And the reason for that is 10 years ago or so, I'd kind of become aware as you read the literature that there are lots and lots of versions. And so it was really an attempt to say, this is the line in the sand, this is where it is, this is what the free drug hypothesis, free drug theory is, and therefore we should try and standardize and then use that as the way to kind of discuss it or refute it or support it, with experimental data.
No, I see, and I guess the biophase being the fluid that bathes the target, right?
Yeah.
And I guess, we often hear the term free drug hypothesis, and I guess you're using now the word theory. I guess why that shift?
So we mentioned this in the paper. So a theory is a formal idea or a set of ideas that's intended to explain something. Unbound drug at the biophase is responsible for eliciting the therapeutic effect, or at least initiating the molecular interaction that does that. A hypothesis is really a possible explanation for a situation or condition, but it's not yet been proved to be correct, the idea of hypothesis testing. But we're using it as a way to explain something, so we felt theory was better.
No, thanks. And I guess as we probably move through this discussion, we'll refer to, other pieces of work that talk about free drug hypothesis as well. So and you've started to talk now about linking drug concentration and I guess pharmacodynamics, and I guess putting that in the context of free drug theory.
Yeah, absolutely. I mean, we work in a world where we make medicines, and medicines are beneficial. They change something in the body to get you back to a state that's more normal, maybe from a disease or enhanced. And so, I think naturally something like the free drug hypothesis is most relevant when it's pointing towards PK/PD. We don't live in a siloed world of drug discovery, drug development, biology anymore, chemistry. It's all linked. And so I think it's useful to have this as a kind of more translational maxim. Oh, and previously there, I mean, in the paper it shows about 7 definitions, and some of them relate the in vitro concentration to, tissue uptake or effect. There's ones that are more distributional. Some are actually related to effect itself, and some to concentration symmetry as opposed to maybe a transporter. So that was another thing, is driving some sort of consistent argument and linking it to pharmacology. PK/PD seemed to be the most sensible thing. It's relevant to all of us then as a maxim.
So yeah, I mean, you mentioned there were numerous definitions in the literature. So I guess this is about, I guess being consistent and in that regard. So maybe we can move on, and I guess let's now turn to some of the fundamental principles that underlie the free drug theory. You in your article you refer to, I guess, a number of principles: law of mass action, Hill-Langmuir, isotherms, drug receptor theory. Maybe we could walk through these, and you could explain what these are, and I guess how they interrelate to underpin this drug action at the target.
So essentially, the law of mass action introduces the notion of rate constants. Say a molecule A moves around haphazardly, randomly, bumping into other molecules and changing direction. If a molecule B is present and A and B can react, and then we know the more times they meet, the more chance there is of them reacting. So the law of mass action is really a formulation of that mathematically. The rate of reaction equals little rate constant K multiplied by the concentration of A multiplied by the concentration of B. And, it's similar for biochemical equilibrium as well. There'll be a forward reaction and a reverse reaction. You might call them K1, K2, K1, K off. And from that sort of little k's, you can get the equilibrium constant, big K, KD, for example. That idea of the law of mass action, it's present in the formulation of Michaelis-Menten kinetics, distributional constants Kp, the total Kpuu. So very valuable there. And then the law of mass action really was the basis for Archibald Hill's work in surface chemistry, the binding of molecules from the gas phase to surface chemistry, and Langmuir and the biology of nicotine acting on frog muscles. But that idea of the law of mass action was a mathematical description of the binding. And from that came the idea of receptor occupancy in the Emax model that we know now. So it's really using law of mass action to conceptualize a mathematical description of the shape of a curve, for example, a receptor occupancy concentration curve. So, and that applies in all places. If you have a mathematical model using rate constants, distributional constants, and concentrations, you're really bringing the law of mass action into action. And free drug theory is the most powerful way to connect that. You have an active concentration in close proximity to the target binding, or strictly, I guess, the molecular initiating event. So from the law of mass action to the kind of isotherms, your concentration effect curves, to kind of drug receptor theory, there's a natural progression, all based on pretty well understood tenets of physical chemistry.
So kind of fundamental principles that govern the kind of movement of substances and their interaction with other substances, I guess. Yeah,
and there's 2 important things in that phrase I just mentioned. Active. In free drug theory, we're referring to unbound, and also the binding of a molecule to its target is a molecular initiating event, if you like. There may be other things downstream that make it easy or not easy to kind of map, but that idea that something bathes the receptor and that binding interaction is based on that concentration, of the free drug.
And I guess what's interesting here, you talked about PD, pharmacodynamics, kind of referring to here, but really here you've perhaps used a key phrase here, that the molecular initiating event. So this isn't some downstream measure. This is fundamental. You're saying, drug action actually at the receptor.
Yeah, and maybe as well, you could have a room of people that have slightly different opinions of free drug theory or free drug hypothesis, depending on what they'd read in the literature, and also your understanding of the mode of action. And maybe that's where sometimes, in project meetings, that one may lead to both of those may lead to confusion, actually. And the other thing we know is that I guess I might be, jumping ahead, and apologies if I am, but obviously in that as well, we know concentrations can be modified by pH, by clearance processes, and by things like carrier-mediated transport. So trying to know what that biophase concentration is the important thing. There may be surrogates, but that's the one that the definition it refers to.
No, thank you. And I think that's probably a useful link perhaps into the next question, which was around, the major processes or that actually impact these local unbound concentrations at the target. And I guess, what actually gets to the receptor or the target.
Yeah.
You talked about pH there. Maybe you could elaborate. And are there other processes we need to be concerned about?
Yeah, so I mean, there are definitions like Dennis Smith's paper where they kind of talk about this sort of thing as well. So if a drug is freely permeable, it will passively diffuse across membranes. So the unbound concentration will move from somewhere near the site of administration to the site of action. Now, along the way, it faces lots of issues, if you like, from a sort of DMPK ADME perspective. The drug may well be cleared. If it's a charged molecule, it may be ionized in different organelles. It's obviously binding non-specifically as well, which is essentially taking it away from, being able to bind specifically. And then you've got carrier-mediated uptake across cellular membranes. So the body is trying to eliminate the drug. We're trying to get it somewhere to make an effect. We're balancing all of those things. So we have to bear that in mind when we're trying to understand, the nature of pharmacology, PK/PD, in light of pharmacokinetics.
And you mentioned another piece of work there by Dennis Smith leading Edward Kearns, I think, is the one you were referring to in Nature Reviews Drug Discovery from 2010. Where they discuss the effects of, plasma protein binding on in vivo efficacy and misconceptions, etc. And also very much talk about the free drug hypothesis, perhaps being a couple of parts to it. I think part one, at steady state, the free drug concentration is the same on both sides of any biomembrane. There are exceptions to that, and maybe we can come on to that in the second part. The free drug concentration affects pharmacological activity. So another, extremely useful article to read about these concepts as well. Scott, you mentioned there non-specific binding, and there's a section in your article on non-specific binding, particularly talking about proteins. I guess non-specific binding to proteins that are not the target of interest. Maybe you could just elaborate a little bit there. And there's a really nice diagram that should refer people to as well, that perhaps draw some comparisons with monoclonal antibodies. Maybe you could just talk through the non-specific binding concepts here.
So proteins, lipids, it could be that as well. So I mean, at small molecules is a good example where we start from, because we're trying to balance the physicochemical properties. We want a target interaction, but we need to get it there. So we have to cross all those biomembranes. We've got clearance to contend with. Permeation is important. So some level of lipophilicity or heteroatoms have to be added to the molecule to make that specific binding and to allow it to traverse membranes, etc. But it's no surprise that a bit of lipophilicity and some functionalization with nitrogen, oxygen, sulfur, etc. means that it binds non-specifically as well. And but that idea is pretty consistent, across all interactions. So when we were trying to formulate the paper, and Dave Fairman was talking to me, actually, he just moved into a new house, and he's electric meter, or well, his electricity had gone. So he was speaking to me in the car on his mobile phone. And he had an engineer in there. And this sort of idea kind of came from there. And I know it was cold because it was just near December. And it was really if you imagine a cytokine. So and the example in the paper is IL-5, but it could be any. So you have IL-5 and it binds to IL-5 receptor, and that leads to pathway stimulation. If you add an anti-IL-5 antibody in there, that binds to IL-5, and so effectively that reduces the activity, it reduces the stimulation. And we can draw the rates for that, K on, K off, etc. Everyone's happy. It's well modeled in the paper that Dave shows. If you think about the thought experiment, if you take the mAb, the monoclonal we've added, and think about that as human serum albumin or something else, the drug is IL-5 and the target is IL-5 receptor, the equations are the same. So when we're thinking about total and free for the free drug hypothesis, it's exactly the same as the kind of rate constants and stuff that we'd look at elsewhere. So there's a really important question of why we've got to be careful as scientists that we don't think something is okay in one place when all the physical chemistry rules are being applied and it's not the same somewhere else. And so somewhere in the paper we mention this is like open-mindedness and curiosity. They're two really, really critical parts about science, as well as we think something might not be right. Can we test it? You know, they're all part of the scientific method. And, you know, part of the issue with this was how do you get it out? And maybe we'll talk about Holmes later and that idea. But so it's a really nice connection to biochemistry.
I thought that was a really nice analogy. Certainly. So maybe, I mean, you mentioned Holmes, and you know, we haven't said already, but you know, Sherlock Holmes and his quotes is a running theme throughout the article. Maybe this is a first time to look at one. Maybe, you know, we should perhaps turn now to the evidence for the free drug theory, Scott. And you know, there's a quote from Holmes which I think you used: "Before we start to investigate, let us try to realize what we do know, so as to make the most of it and to separate the essential from the accidental." So, you know, there's a section about evidence for the free drug theory.
James Yates, actually, I think he was the one in the conversations that said about Sherlock Holmes, and there's an earlier quote about as well, and that just helped unlock everything, because Holmes was really a good mirror for the scientific method in general, the way he applied stuff. So I hope people enjoy that bit. It's funny. I mean, I guess one of the questions is, you know, for me, thinking about the free drug hypothesis is, how does a molecule non-specifically bound several angstroms away from the target impact drug effect? And maybe we can come back to that. We were kind of talking about examples, I think, weren't we, of where the free drug hypothesis might be applied. Yeah, so there's some good examples, and there's a few for BBB. I know those better,
Blood-brain barrier,
because the neuroscience was the bit I was in. Yeah, the blood-brain barrier. Sorry. And there are some mentioned in oncology about VEGFR2 receptors and in antibiotics as well. But one of the ones that we mentioned in the paper was from Jeanette Watson on sort of D2 antagonist binding, marketed drugs, different affinities for the D2 receptor, some subject to PGP efflux like risperidone, and some passive across the BBB like saprazidone, and some that appear to have uptake like clozapine and chlorpromazine. And if you looked at KPUU after the fact, because KPUU didn't exist as a concept then, but if you look at their KPUUs, like the ones that's the unbound concentration gradient across the BBB. So risperidone much less than 1. If it's passive, KPUU is about 1, and if it's uptake, KPUU is greater than 1. And if you look at the sort of concentration receptor occupancy curves, they overlay and only overlay when you account for the differences in the affinity of those drugs at D2 and also the unbound brain concentration. And that's just the concentration occupancy curves effectively merge, overlay one another. And that's a really nice example.
No, and it's interesting that you talked there about having perhaps having uptake, and we previously mentioned about unbound concentrations being the same across the biomembrane. That's probably an example where that isn't the case. However, I think what you're saying is it's still the unbound concentration at the target that drives the effect. It's just that unbound concentration may not equate to actual, say, unbound concentration in plasma.
Yeah. I mean, I was reflecting on an example of a program that I remember working on, and it was a neurodegeneration target inhibitors. They were using a mouse model, chronic dosing, so you'd assume it's steady state, and they took a behavioural endpoint, and then we got blood-brain. We had plasma-free fraction, so we could measure the unbound plasma concentration, and that was for many compounds that are in that series. And this is pre-KPU days, so we didn't have unbound brain. There was a general trend across the compounds, but it was pretty variable, and it was tighter when you kind of looked at the effects with total brain than unbound plasma. And I can remember the program leader, Ph.D. We're not slouches, kind of convinced total brain was more relevant. And I reflect back on that, and I wonder whether we had different opinions of what the free drug hypothesis meant. I think, for that individual, he probably was thinking unbound plasma is the same as biophase concentration. I was thinking of the biophase concentration. The drugs weren't particularly permeable. They were PGP substrates. So, in my mind, it was already modulated. But it's a very, and then the mechanism would be pretty complex as well. And we have one endpoint and one time point per compound. So, there's all these things in the mix. But fundamentally, if we had a different definition, we would never agree, and we would always think something else.
It seems in that example as well, the endpoint is probably several steps downstream from actual target interaction.
Yeah, and the other thing when you think about that example as well, and we might come on to the ones where you refute the free drug hypothesis, is for a poorly permeable compound where KPUU is not one necessarily in all the tissues and biomembranes, so we know the concentrations are being modulated. So even if something like non-specific binding, which may be high capacity, low affinity, is allowing the drug to become almost a reservoir, the total drug become a reservoir near the target site, maybe faster than the kinetics of the drug moving across the biological membranes. Well, then the free drug hypothesis still applies. We just haven't factored in the rate constants in the right way. We've missed something. If there was a conformational change because of the presence of, or something, I don't know, that where the nearby drug several angstroms away is affecting the target, then no, the free drug theory doesn't apply. But we have to kind of have an idea. We can't just say, personally, total is more relevant, because that has massive implications for the kind of molecule that you're selecting, and also the kind of we spend a lot of time trying to factor out non-specific binding when we measure affinities, KDs. So are you measuring the right thing in the first place for your lead optimization? You don't hear anyone say, oh, maybe the permeability I've measured, we need to factor in the non-specific binding to get the right value.
Your comment there, reminds me of another Sherlock Holmes quote used in the article. Circumstantial evidence is a very tricky thing. It may seem to point very straight to one thing, but if you shift your own point of view just a little, you may find it pointing in an equally uncompromising manner to something entirely different. Yeah. Scott, maybe we move on to talk about, talk about unbound drug concentration, and being so important. How do we get to a representative measurement of what that unbound concentration is at the target? Referred back to Dennis Smith's article earlier around the unbound concentration being similar across the biomembrane, and you've talked about measuring unbound concentration in plasma, and that's clearly a very available matrix. Is that a good place to start?
Well, we know yes, we know that we have assays that can look at things like passive permeability. We have a good array of transporter assays that's always growing. So we can kind of get a good view across different carrier-mediated systems, whether they impact the disposition or not, the concentration gradient. We can measure Kas, pKas, pKbs. We know to know the ionization state. If our target's in a certain organelle with a different pH, then, so mathematically we may be able to do it, and we can get an idea of passive or not passive. Unbound plasma is a good place to start. If you're seeing something that looks a bit odd, then, we may well be already generating data that allows you to think about, well, is the unbound plasma concentration really representative of what's going on, at or close to the target in terms of unbound concentration.
But and you say that, but I guess also with the complexities of the mechanisms and the potential mechanisms at play here, there could actually equally be perhaps something that isn't understood at the time. Yeah,
I mean, particularly in drug design, early drug discovery, we're still in a very, very sort of nascent stage of that molecule. You don't have that molecule's development, and there are a couple of examples in there, three-ish, of where people have questioned whether total is more relevant than free. And there's one with there's one where when you look at the mechanism and the binding interaction occurs, then that's translocated across a membrane, which then initiates a reaction. So, I think the thing about the free drug hypothesis is you can take it and say, okay, free plasma doesn't appear to work. Can we estimate or measure something else, mathematically with data that we've got, or is there a way of getting closer? And then we can also question the mechanism of action very early. It's all about applying the scientific method and not making assumptions.
And I guess that's the concentration side of things. There's also the potency side of things as well, with the molecules interaction to the target. Project teams are often in situations where there is this need to translate potency across various different assays, particularly from in vitro to an in vivo situation, to understand those relationships that you've described of target binding and efficacy. And these mechanisms, are kind of often pretty complex, but there's clearly a need to understand this well and relate unbound concentrations to also the effect over time. I mean, are you able to elaborate on any of the concepts here where we may see differences between in vitro and in vivo systems?
Well, one of the things that we mention in the paper is if you've kind of got observations that contradict maybe the free drug theory, and the paper, just as a reminder, doesn't say the free drug hypothesis applies. What it says is it's a good place to start, and then if you don't see the correlation, what's that telling you? Is it informing you about something that you don't know? So is there an energy driven distributional or clearance asymmetry that's altering the free concentrations? So KPUU isn't one. Is the system at steady state when the measurements are made in vitro or in vivo? Could your understanding of the MOA be incomplete? What assumptions have you made for the experimental conditions? Like, does in vitro truly reflect in vivo? And then, can you really make an assumption under the error of the measurement? And I think that was one of the problems with the example we had there, is the data was variable when we were looking at that neuroscience project. So you needed a dash of objectivity to really say whether you.
I know Dennis Smith's article as well also refers to some of those differences which I think are important here, aren't they? In vitro assay systems often contain protein, of course, and that may not be that may affect the assumption of what the unbound concentration at the target is. I know in my experience that's made a substantial difference actually in some projects.
Yeah, absolutely. Absolutely. It's we have to be able to, if we see something anomalous from a starting point, it gives us the opportunity to challenge what we understand. That piece of curiosity and open-mindedness is the bit that kind of allows us to really find the good, well-precedented, good drugs that will become well-precedented targets and, medicines for the future.
And I guess also, the expression of the target in vitro cell systems as well, and whether that is, truly representative of in vivo. Yeah. So I guess, we talked all the time. This is all about, I guess, getting an understanding, but also about designing better drugs in inverted commas. How can projects, I guess, maximize the free drug concentration at the target? Are there particular properties that teams should be focusing on?
Yeah. I mean, there was a nice article by Dennis Smith and Malcolm Rowland. It pointed out something still that's really important and relevant, is that,
this is one in drug metabolism and disposition, Scott. Yeah.
Yeah, that selecting drug candidates with good lipoidal permeability remains an important maxim for molecules to cross membranes. If you don't have that, your KPUU will be much lower than 1, or if it isn't, it's just going to take a long time to get to steady state. So that's an important one. Obviously, we can measure KPUU from slices and homogenates and stuff now.
Mm-hmm.
And there's been some interesting papers, about regional variations in KPUU in the brain. Irena Lorien's published some of that with her group, Margareta's group, where they've used MALDI and a mixture of sort of unbound homogeneous measurements to show, how you might have local differences in unbound concentration. So, imaging techniques are becoming more sensitive, more important, faster. They can work at the kind of rate of the lead up cycle. So, we live in a very advancing age technologically. We're very lucky. When you look back at some of those early papers, they didn't even know molecules or whatever truly existed. They didn't know about receptors and all that sort of thing. We can probe at a level so refined, and that's only going to get better.
And I guess, would you agree that even some of the parameters that we can optimize from the traditional assays are important here? Reducing metabolic stability, I guess, of course, improves unbound systemic exposure, and you mentioned before, reducing efflux, I guess, if that's an issue at the tissue barrier.
Yeah, I mean, the thing new technologies, new measurements are going to help, but a drug has to be stable, right? It can't be chewed up so much that it can't get to the site of action. So they're all really, really valid. It's how we take those and cross the chasm to understanding PK/PD and not forgetting the principles that allow drugs to bind to targets to elicit an effect. The initiating event, the unbound concentrations, how they're linked at steady state and temporally, the complexity of that reaction to the biochemical reactions that then ensue to whatever we measure.
Thank you, Scott. I mean, how do you reflect on this topic now? Where do you see this, I guess, going in the future?
So I hope the paper goes some way to standardizing the kind of idea of what free drug theory is, so that we don't have scientists sitting at the table with completely different opinions of what that definition means. I think that confusion will make it much easier for project teams and scientists to talk head to head at the same lingo. And I think the idea of trying to kind of link it to the PK/PD side makes it much, much more powerful. That's what we're here for, to develop drugs. And drugs is efficacy.
Thank you.
Yeah.
Thank you, Scott. I guess there's one final Sherlock Holmes quote, which I think is worth saying here, very pertinent to this topic. It's an old maxim of mine that when you have excluded the impossible, whatever remains, how improbable must be the truth.
No, absolutely. And I have a couple of people just say thank you to Margareta and Irena Lorien. They offered me the chance to write the paper. And then Dave Fairman and James Yates, who with the discourse and the laughs along the way were really great. And it allowed me to free that worm in my brain, if free is, pardon the pun.
Scott, it's been a fascinating and insightful discussion. Thank you very much for your time. Before I close this, I'd like to provide just reference to the core articles we've discussed in today's podcast. As a reminder, they're the one from Summerfield, Fairman and Yates. Free drug theory, no longer just a hypothesis in pharmaceutical research, from 2022. Dennis Smith, Lee D and Edward Kearns, the effect of plasma protein binding on in vivo efficacy, from Nature Reviews Drug Discovery in 2010. And the final one was Dennis Smith and Malcolm Rowland, intracellular and intra-organ concentrations of small molecule drugs, the theories and uncertainties in infectious disease and oncology. And that was from Drug Metabolism and Disposition in 2019. So thank you for taking the time to listen today, and look out for more podcasts in our Pharmaron DMPK Insights series. And of course, there's our DMPK webinar series available online as well, covering a variety of key questions relating to DMPK science in drug discovery and development. Bye for now.